Good morning, students. In this lecture, we are going to discuss about Mendel's experiments on inheritance, monohybrid cross, dihybrid cross, Mendel's law of inheritance, and its applications. So, at first, Mendel means G.J. Mendel, or you can say Gregor Johann Mendel, who was the father of genetics, and he was the first person who studied bio in combination with maths, and therefore he was considered as father of genetics. He contributed a lot in inheritance or study of genetics. His research material, he has selected Pisum sativum or garden pea plant as his research work. There are a number of reasons for the selection of garden pea as research material by Mendel. So, reasons for the selection of garden pea by Mendel. There are three important reasons due to which Mendel selected garden pea plant for his research work. Number one, it has 7 pairs of contrasting characters or features. Number two, pea plant is a self-pollinated plant, and also artificial cross-pollination can take place in it, and also artificial cross-pollination can take place in it. And number three, they are always found in pure form. So these are the most important features. They are found in pure form. One more reason, it can be grown around the world as a herbaceous plant. So it takes very less time to harvest. So these are the 4 most important reasons for what Mendel selected garden pea as his research work. It has 7 pairs of contrasting characters. Number 2, pea plant is a self-pollinated plant, and also artificial cross-pollination can take place in it. Number 3, they are found in pure forms. And number 4, it can be grown around the world. So these are the 4 important features. Now there are 7 contrasting characters are found in seven pairs of contrasting characters which are found in pea plant, and these are character means here feature, dominant trait, and recessive trait. So here character, dominant trait and recessive trait. As you know, dominant trait means the trait which is expressed in F1 generation, and these are very common. Recessive trait means the trait which is unexpressed in F1 generation and is not so common. Number 1, height of the plant. Height of the plant. Here tall plants are dominant trait, tallness, and dwarfness. Dwarfness is the recessive trait. Number 2, seed color. Usually colors of pea are yellow, whereas some varieties of pea seeds are green in color. Seed shape. Mostly seeds are round, whereas some are wrinkled. So wrinkled is here recessive trait. Number 4, pod shape. So pods are mostly inflated or smooth, whereas some are constricted. Number 5, pod color. Mostly the pods of pea are here. Number 4, pod color. Pod color are green mostly, but very few are yellow. Number 5, pod shape. Mostly these are inflated or smooth, but very few of them are constricted. Number 6, flower position. Axillary, and some are terminal. So terminal. And last point, contrasting characters. Here flower color. Mostly these are purple or violet, but some varieties have white colored flowers. So these are the 7 pairs of contrasting characters. Here height of the plant, tall plants are dominant whereas dwarf are recessive. Seed color yellow and green. Seed shape round and wrinkled. Pod color green and yellow. Pod shape inflated or smooth and constricted. Flower position axillary and terminal. Flower color purple or violet and white. So these are the 7 pairs of contrasting characters in pea plant, and therefore it is one of the most important feature or reason for the selection of this plant by Mendel. There are 2 different types of characters. These are dominant trait and next one is recessive trait. So dominant trait are the traits which are expressed in F1 generation. And it can be expressed in F2 generation, but it is expressed in F1 generation. These are very common. And second one is recessive trait. The trait which are unexpressed in F1 generation in the presence of dominant trait, and this trait are not common. So in this way, here dominant trait which are expressed in F1 generation and recessive trait which are unexpressed in F1 generation and all, but in F2 generation it is expressed. Now, monohybrid cross. Monohybrid cross. When a cross is made between one pair of contrasting characters or features, then it is known as monohybrid cross. Here only one feature is taken for consideration, under consideration. So monohybrid cross, we are taking an example of monohybrid cross. We are taking tall plant and dwarf plant. Now, dwarf plant and tall plant and dwarf plant, these are the 2 traits of the same character, height of the plant. So here character is height of the plant. Traits, there are 2 traits, tall and dwarf. Again, genes responsible for these capital T and small t. Alleles here capital T and small t and small t. So these are the character, trait, genes and alleles. Now capital T, it is a pair of genes for tall plant, and small t, it is a pair of alleles for dwarf plant. Now gamete formation take place, here capital T and here small t. So these are the gametes. Here tall plant, suppose it is male plant, and dwarf plant it is female plant. Now crossing is made here. Crossing means here cross pollination. Crossing is made and here capital T small t type of zygote forms in F1 generation, and it is known as all heterozygous tall plants appear in F1 generation, first filial generation. So it is the genotype and this one is the phenotype. Here capital T and small t, capital T small t, it is genotype for F1 generation, and phenotype here all tall plants, it is phenotype. Now in the next generation means F2, when a capital T is crossed with capital T small t, capital T small t is crossed with capital T small t, it is shelfing. Shelfing means shelf pollination. Then we have to make a Punnett square. It is a Punnett square, and here male gametes capital T and small t, and here again capital T small t, and these are the female gametes. After crossing capital T, here capital T small t, again here capital T small t, and here small t, small t. These are the phenotypes we have written. Now these are the genotypes, and now phenotype we are writing here. Capital T, it is pure tall plant. Here it is hybrid or heterozygous tall plant. Here again hybrid or heterozygous tall plant, and here dwarf plant. It is homozygous recessive condition, and here it is always pure. Now there are two ratios obtained here, and these are known as monohybrid ratio. There are two different types of ratios obtained for F2 generation, and these are number one phenotypic ratio. Phenotypic ratio means here only tall and dwarf plants obtain here. Tall plant 1, 2 and 3. These are the three tall plants. So here 3 and only one dwarf plant, and therefore 1. So it is the phenotypic ratio for F2 generation. And genotypic ratio. Genotypic ratio here capital T is to capital T small t is to small t. Here you can see capital T 1 is to capital T small t, here 1 and 1 2, and again small t 1. So it is the genotypic ratio for F2 generation. Here monohybrid ratio for F2 generation. So in this way we can make a cross between one pair of contrasting character, and we can obtain genotypes, phenotypes, and also phenotypic ratio and genotypic ratio. Now phenotype for F1 generation, all heterozygous tall plant, means tall plants you can simply say. And genotype capital T small t, means it is the heterozygous condition. So in this way, this is one example of monohybrid cross, and we can obtain phenotypic ratio and genotypic ratio for F2 generation. Now what will be the phenotypic and genotypic ratio for F1 generation? Here all heterozygous tall plants are obtained. It means the ratio for genotype as well as phenotype is 1:1 or 4:4. You can simply write 1:1. So here it is the genotype and this one is the phenotype of F1 generation, and here genotypes and phenotypes for F2 generation and phenotypic ratio and genotypic ratio. So you must remember monohybrid ratio, phenotypic ratio tall is to dwarf is to 3:1 and genotypic ratio 1:2:1. Now dihybrid ratio, dihybrid cross. Dihybrid cross is a cross which is made between two pairs of contrasting characters. Dihybrid cross. It is a cross made between two pairs of contrasting characters. Contrasting characters are taken here for study. Now we are taking an example. Here we have to take two pairs of contrasting characters. Suppose seed shape of seed and seed color. It is taken under consideration. So in flat round and wrinkled, these are the two traits of the same character, shape of seed and seed color, here yellow and here green. These are again two traits. Here capital R, capital Y, and here small r, small y, small y. There is a rule in genetics. We express dominant trait by the first capital letter of the trait. Here suppose round, capital R, we have to take as to express the genes or alleles. Here wrinkled starts with W, but we can't take it as expression. We have to take a small letter of the same alphabet. So here capital R is taken as dominant trait. So small r is the is taken as expression for the wrinkled. So in this way we take, and in F1 generation we found capital R small, capital R small r, capital Y small y type of zygote, and it is round and yellow seeds obtained. So it is phenotype, and this one is the genotype for F1 generation. In this way we obtain genotypes and phenotypes for F1 generation. Now possible combinations for F2 generation. Here, by making a Punnett square, we can obtain here capital R. These are the two, capital R, small r, capital Y, small y. These are the gametes. Now, capital R, capital Y, capital R, capital Y, capital R, small y, and here capital R, capital Y, capital R, small r, capital Y, and small r, small y. These are the possible combinations or gametes for F2 generation. Now we take these type of gametes for study of F2 generation for the same. Here F2 generation, capital R small y, R capital Y small y is crossed with capital R small y, small r capital Y small y. Here shelving is made. Here Punnett square is made, in which 16 boxes are there. Now male gametes, capital R, capital Y, small r, capital Y, capital R, small y, and small r, small y. In the same way, we have to write here all these combinations or gametes. Here it is female gamete. Now we have to cross them. Now we have to write phenotype. Here round and yellow, round and yellow, round and yellow, round and yellow. Here again round and yellow, wrinkled and yellow, round and yellow, wrinkled and yellow, round and yellow, round and yellow, round and yellow, round and green. Here again green, round and yellow, wrinkled and yellow, round and green, and here wrinkled and green. So in this way we obtain round yellow, wrinkled yellow, round green and wrinkled green. So these are the 4 different types of phenotypes we obtained. Here round yellow 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8 and 9. So here 9, wrinkled yellow 1, 2 and 3. Now round green are here, round green 1, 2 and 3, and wrinkled green only 1. In this way it is the phenotypic ratio for up to generation or for dihybrid cross, and it is 9:3:3:1. So in this way we can obtain different genotypes and phenotypes for up to generation in a dihybrid cross, and here we obtained phenotypic ratio also. So in this way these are the monohybrid and dihybrid crosses we studied, and after it we study about Mendel's law of inheritance. Mendel's law of inheritance. Mendel studied about monohybrid and dihybrid crosses by taking different pair of contrasting characters and formulated some laws, and it is known as law of inheritance. Mendel's law of inheritance. Mendel's laws of inheritance. Number 1, law of dominance. According to this law, when one pair of contrasting character is studied, then one trait expresses itself in F1 generation and the second trait can't express itself. Means expressed character is known as dominant trait, and unexpressed character in F1 generation is known as recessive trait. Now second, law of segregation. Law of segregation, which is known as law of purity of gametes. Law of purity of gametes. Law of segregation or law of purity of gametes. According to this law, out of 2 traits or alleles of a pair of genes or 2 traits of a character are separated and inherited to the gametes. They cannot blend with each other and they are always found in the pure form, and therefore law of segregation is also known as law of purity of gametes. And number 3, law of independent assortment. According to this law, when two pairs of contrasting characters are crossed and studied, then one type of trait or allele are independent to inherited in the gamete. They cannot depend on the other trait for its inheritance. So you can say when there are two pairs of contrasting characters, the distribution of the allele of one character into the gametes is independent of the distribution of the alleles of the other character. And in this way, these are the three laws or Mendel's laws of inheritance. Thank you, students.